Hello students, Jinali Shah welcomes you all on behalf of Saraswati International Academy. Today we will begin with the next part of the chapter Biomolecules. Students, in the previous video we have studied regarding the biomolecule carbohydrates. In today's lecture we will begin with our next biomolecule, and the name of the biomolecule is the nucleic acids. Now students, we know that there are two basic types of the nucleic acids present in the living cells. One of it is DNA and another one is RNA. So students, first of all, what are these nucleic acids made up of? We know that nucleic acid is a macromolecule, and this macromolecule is made up of the monomers called as the nucleotide. So we can say that the nucleic acids are the polynucleotides. How can we define nucleic acids? They are considered as the polynucleotides. Now what are nucleotides? How it is formed? We will study that. So we know that there are two major nucleic acids. One is DNA and RNA. And basically it is the DNA which acts as the genetic material in all the living organisms. Except for the few viruses, it is the DNA which acts as the genetic material. It means it stores the information. And RNA basically will be involved in the expression of the information. RNA basically copies the information from the DNA, and then the genes will be expressed in the presence of the RNA. So now, how this DNA and RNA are formed? They are nucleic acids. Nucleic acids are the macromolecules, so they will be formed of the monomers, and the monomers of the nucleic acids are called as nucleotides. So what are nucleic acids? Basically nucleic acids are polynucleotides. So students, the other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is nucleic acids. These are polynucleotides. Poly means many. So when many nucleotides are joined, it forms the nucleic acids. Together with polysaccharides and polypeptides, this comprise the true macromolecular fraction of any living tissue or cell. So macromolecular fraction may, along with polysaccharide and polypeptide, the other component present is the nucleic acids. For nucleic acids, the building block or monomer is nucleotide. What are nucleic acids? They are polynucleotide. So what is a monomeric unit of nucleic acid called as? It is called as a nucleotide. They are the information molecules of the cell. So basically they store the informations of the cell. They also take part in energy transfer system of cells. Energy transfer system, it means which are involved in the production of energy, generation of energy, like ATP. We know that ATP is the energy currency of cell. So this ATP is nothing but it is a nucleotide. So nucleotide, one type of a nucleotide is ATP involved in the energy transfer mechanism. Nucleic acid was first isolated in 1868 by Swiss physician Friedrich Mischer. So who first isolated nucleic acid? It was Friedrich Mischer. He found out the weakly acidic substance of unknown function in the nuclei of human white blood cell or pus cell and named this material nuclein. So what Friedrich Mischer found? He found some weak acidic substance in the nucleus of the human blood cell or pus cell, and he named that acidic substance. He didn't know the function of that particular acidic substance, but he found that yes, it was the acidic substance, and he gave the name nuclein to that acidic substance found in the nuclei of the human white blood cells. In 1884, Oscar Hertwig, he wrote that nuclein is the substance which is responsible for transmission of hereditary characters. Okay, 68 में Friedrich, he found the acidic substance in the nuclei of the white blood cell and he named it nuclein. In 1884 what Hertwig wrote? He wrote, उन्होंने सिर्फ लिखा, he didn't prove anything but he wrote that nuclein is a substance which is responsible for the transmission of the hereditary character. उसने क्या बोला कि nuclein वो substance है which transmits the hereditary characters from generation to generation. This nuclein was renamed as nucleic acid by Altmann in 1889. So, nuclein was later named as nucleic acid by Altmann in 1889. He discovered the existence of two types of nucleic acids. Altmann, he said, or he discovered that there are two types of the nucleic acids present. Today we know that nucleic acids, one is RNA and another one is the DNA. That is ribonucleic acid and deoxyribonucleic acid. Meischer, he separated the nuclein into protein and nucleic acid component. Later माइशर ने क्या किया कि जो न्यूक्लिन उसने डिस्कवर किया था उसको सेपरेट कर दिया, किसमें? प्रोटीन कंपोनेंट्स एंड न्यूक्लिक एसिड कंपोनेंट्स में. द न्यूक्लिक एसिड्स वर फाउंड टू बी मेजर कंपोनेंट्स ऑफ क्रोमोसोम्स, द स्मॉल जीन कैरिंग बॉडीज इन द न्यूक्लियाई ऑफ द कॉम्प्लेक्स सेल. So nucleic acids, they are the major component of chromosomes and these chromosomes are the small gene carrying entities in the nuclei of the complex cell. So what are nucleic acids? They are polynucleotide. So let us understand what is nucleotide and what is it composed of. So let us begin with the nucleotides. So they are the compounds of carbon, hydrogen, oxygen, nitrogen and phosphorus. What nucleotide contain? They contain carbon, hydrogen, oxygen, nitrogen and phosphorus. Now, a molecule of nucleotide itself is made up of three smaller molecules. जो nucleotide है वो खुद 3 small molecules से बना हुआ है. So, a molecule of nucleotide consists of 3 smaller molecules of different type. So, nucleotide is made up of 3 small different types of molecules. Which are they? One is phosphoric acid which occurs in the form of the phosphate group. The phosphoric acid which occurs in the form of the phosphate group. Then a 5 carbon or pentose sugar molecule. So, here it consists of a 5 carbon or a pentose sugar, monosaccharide sugar, and that sugar either will be a ribose sugar or it will be the deoxyribose. दो टाइप की शुगर है, one is ribose and another one is deoxyribose. अगर RNA है which is ribonucleic acid, उसमें ribose sugar होगा, and DNA which is deoxyribonucleic acid, so उसमें deoxyribose sugar होगा. And the last component is a nitrogenous base. There are 5 different types of nitrogenous bases. We will study them. So a nucleotide, it is composed of how many components? 3 smaller components. Which are those 3 smaller components? One is phosphoric acid which occurs in the form of phosphate group, a 5 carbon or a pentose monosaccharide sugar, that is either ribose or deoxyribose, and there is a nitrogenous base. We will study each component in detail. So first of all, phosphoric acid. The formula for phosphoric acid is H3PO4. It is a biologically called as phosphate group. So phosphoric acid biologically is called as phosphate group, and this phosphate group arises as PO4 minus ion. It occurs as PO43 minus ion. Okay. Now, a nucleotide may have a 1, 2 or 3 phosphate group. न्यूक्लियोटाइड के अंदर एक फॉस्फेट ग्रुप भी रह सकता है, 2 भी रह सकते हैं, या मैक्सिममली 3 phosphate groups will be present. When we talk about ATP, the ATP consists of the 3-phosphate group. It is adenosine triphosphate. The phosphate group gives acidic nature to the nucleotide. So basically nucleic acids acidic है. Acidic क्यों है? हम बोलते हैं nucleic acid. So basically the acidic nature is due to the presence of the phosphate group. So this is regarding the first component, that is the phosphoric acid. Now the next component is the pentose sugar molecule. Which sugar we are talking about? Pentose sugar. Penta, it means 5. A sugar containing how many carbon? 5 carbon. That sugar is called as the pentose sugar. So the sugar molecule in nucleotide may be ribose, whose formula is C5H10O5, or it may be deoxyribose, whose formula is C5H10O4. वेरी वेरी इंपॉर्टेंट, ये दोनों में से ही कोई भी एक शुगर प्रेजेंट होगी. अ न्यूक्लियोटाइड में कंटेन आइदर अ राइबोज शुगर और इट में कंटेन अ डीऑक्सीराइबोज शुगर. नाउ हमने जब कार्बोहाइड्रेट स्टडी किए तो वी हैव स्टडीड दैट उनका स्ट्रक्चर स्टडी किया था. तो बेसिकली हमने चेन स्ट्रक्चर स्टडी किया था. बट दिस कार्बोहाइड्रेट्स में ऑल्सो अकर इन द रिंग फॉर्म. The carbohydrates, these sugars, may also occur in the ring form. So when we talk about the pentose sugar in the nucleotides, always they occur in which form? Ring form. So both the sugars are in furanose or pentagon state. Furanose or pentagon, that is, it is a 5-ringed structure. Here you can see the diagram. This is a deoxyribose sugar and this is a ribose sugar. Here you can see the ring shape. So ये ring में there are 4 carbon and 1 oxygen. It is a pentagon ring. So how many corners will be there? 5 corners. Of the 5 corners, 4 are occupied by carbon. See students, first, second, third and 4. So 4 corners are having the carbon. And 1 corner is occupied by what? It is occupied by oxygen. Now बचा क्या है? 5th carbon बचा है. So this 5th carbon, it occurs outside the ring along with the molecule of H2 and OH. So this is a structure of the ring form of the deoxyribose and ribose sugar. So both the sugars, this is a furanose ring. Furanose ring या pentagon ring. क्या बोल सकते हैं? Furanose ring or pentagon. It means there are 5 corners. Of the 5 corners, the 4 are occupied by carbon. Here you can see in both the sugars, 4 are occupied by carbon. 1 position is occupied by oxygen and the remaining 1 carbon occurs outside the ring along with the H2OH molecule. The 5th carbon along with H2OH occurs outside the ring. Now see students, structure देखो, very very important. In both the sugars, this represents the first carbon. This represents the second carbon. This position represents the third carbon. This position represents the 4th carbon, and to this 4th carbon CH2OH group is attached representing the 5th carbon. Okay, बाकी सब जगह पे oxygen, OH group and hydrogen, then hydrogen and OH, hydrogen and OH, hydrogen and then OH. Okay. Now ये दोनों का difference देखो. The second position. The second carbon in ribose sugar have the OH group, whereas the second carbon in the deoxyribose it have only hydrogen. It means उसमें 1 oxygen is less in the deoxyribose, and hence it is given the name deoxy. D means removal, oxy means oxygen. So it contains 1 less oxygen atom than the ribose sugar. The second position in the ribose sugar, it consists of the OH group, whereas in the deoxyribose it will only contain the hydrogen. It lacks 1 oxygen. Here you can see this is a second position of the carbon in ribose occupied by OH. In Deoxyribose only hydrogen. It means the deoxyribose contains one less oxygen and hence it is given the name deoxyribose. So these are the pentose sugar molecule which occur in the nucleotide. Now the last component of a nucleotide is a nitrogenous base. So these are the organic bases of the nucleotide, which either is a purine or it is a pyrimidine. So जो organic bases nucleotide में होते हैं, they either will be a purine or it may be a pyrimidine. Now what are purines? What are pyrimidines? The purines and pyrimidines, they are fat heterocyclic compounds having nitrogen and carbon in the ring structure. So basically what are purines and pyrimidines? They are the ring structure. Okay, they are cyclic rings. What are they? They are cyclic ring structure. But which type of rings are they? Heterocyclic. What does that hetero means? Different. यानी जो रिंग के कॉर्नर्स है दे आर ऑक्यूपाइड बाय द डिफरेंट कंपोनेंट्स, यानी some corners are occupied by carbon and some corners are occupied by the nitrogen. So what are purines and pyrimidines? They are flat heterocyclic rings, each having the nitrogen and carbon in their ring structure. Hetero, it means different. Cyclic, they occur in cyclic manner. So that position of the, or basically the corners of the ring, they are occupied by both carbon and nitrogen. Some corners occupied by carbon, some corners occupied by nitrogen, and hence it is called as the heterocyclic ring. Now why it is called as a nitrogenous base? Nitrogen in the rings, they tend to take up H plus ion from the solution and gives the molecules their basic nature, hence the name nitrogenous base. अभी ये रिंग में जो नाइट्रोजन होता है वो क्या करता है? From the solution they take up the H plus ion, okay, and when anything accepts the hydrogen it becomes a base. So as the nitrogen, it takes up the H plus ion from the solution, it gives the basic nature to this component and hence they are called as the nitrogenous base. Now what is the difference between a purine and a pyrimidine? So a pyrimidine molecule is a smaller molecule and it has one 6-membered ring. Very important student. Pyrimidine, it is smaller and it have only one ring, and that ring is how many how many membered ring? 6-membered ring. Now of these 6 members, the position 1 and 3 are occupied by the nitrogen. So 1' and 3' position of this 6-membered ring is occupied by what? Nitrogen, and remaining are occupied by carbon. So we will see its structure. What is a pyrimidine? It is a smaller structure which have one 6-membered ring. Of this 6-membered, position 1' and 3' they are occupied by the nitrogen, and the remaining will be occupied by what? The remaining will be occupied by the carbon. Okay. Now a purine. A purine molecule is larger and it has 2 rings. Very important, the difference between purine and pyrimidine. Pyrimidine में हमने देखा कि there is only one 6-membered ring, whereas a purine molecule is a larger molecule and it have how many rings? It have 2 rings. One is 6-membered ring which is joined to a 5-membered ring at 4' and 5' position. So one is a 6-membered ring. वो जैसे ही pyrimidine की थी सेम रिंग रहेगी कि जहाँ पे 1 and 3 पोजीशन पे नाइट्रोजन थे एंड बाकी सब कार्बन थे. Now this 6-membered ring at position 4 and 5, this 6-membered ring at position 4 and 5 is joined with a 5-membered ring. So एक 4-membered ring है and एक कौन सी membered ring है? 5-membered. बोथ दिस रिंग्स आर जॉइंट. सो उनका जॉइनिंग पोजीशन कौन सा है? 6 मेंबर की जो 4th and 5th पोजीशन है वहाँ पे 5 मेंबर्ड रिंग जॉइन हो जाएगी. ओके, सो नाउ इट हैव 2 रिंग, वन इज 6 मेंबर्ड रिंग. सो जो 6 मेंबर्ड रिंग है उसका नाइट्रोजन का पोजीशन 1 and 3. Along with it, it also carries the nitrogen at position 7 and 9. So there are 4 nitrogens in a purine molecule. They are occurring at position 1, 3, 7 and 9. Purine is a larger molecule with 2 rings. One is a 6-membered ring which is joined to a 5-membered ring at 4 and 5 position. In this complete structure, position 1, 3, 7 and 9 are occupied by the nitrogen. Whereas a pyrimidine molecule is a smaller molecule which contains only one 6-membered ring with nitrogen at 1 and 3 positions. See students, here you can see a difference between a purine and pyrimidine. This is a pyrimidine having how many rings? It is having only one ring with nitrogen at position 1 and 3. Remaining you can see that they are having a carbon, whereas purine is having 2 rings. This is a 6-membered ring and this is a 5-membered ring, and these rings are joined at the position 4 and 5. Nitrogen occurs at position 1, at position 3, at position 7 and position 9, and remaining all are occupied by the carbon. So this is a structure of a purine. This is pyrimidine consisting of only one 6-membered ring, whereas a purine consisting of 2 rings, one is 6-membered and another one is 5-membered. Now we are talking about the nitrogenous bases. Either they are purines or they may be pyrimidines. Now there are 3 important pyrimidines. कितने important pyrimidines है? 3. Which are they? Thymine, whose short form is written as T, cytosine C and uracil U. And out of the purines, there are 2 important purine. By mistake it is written 3. Basically there are 2 important purines. One is adenine represented by A and guanine represented by G. So कितने pyrimidines है? 3. Thymine, cytosine and uracil. And purines कितने हैं? 2. One is adenine and another one is guanine. Now हम जब DNA and RNA की बात करते हैं, सो एक डिफरेंस तो उनका शुगर में रहा. DNA में इज डीऑक्सीराइबोस शुगर and RNA contains the ribose sugar. The second difference between second chemical difference between the two is दोनों में प्यूरिन सेम है, यानी both of them will contain adenine and guanine. Okay. Out of the pyrimidines, cytosine is common for both, यानी दोनों में cytosine भी है. अभी difference rise in one of the component. Thymine is present only in the DNA and uracil is present only in the RNA. So this is another major difference between the chemical composition of DNA and RNA. DNA में कौन सी शुगर है? डीऑक्सीराइबोस शुगर है. RNA में कौन सी शुगर है? राइबोस शुगर. दिस इज वन मेजर डिफरेंस. दूसरा डिफरेंस, adenine, guanine and cytosine, they are common for both DNA and RNA. The difference lies in the pyrimidine, thymine and uracil. Thymine occurs in DNA and uracil it occurs in the RNA. So this is the major difference between the DNA and the RNA. Now see students, here you can see the ring structure of all the purines and pyrimidines. Purine, first carbon, third carbon, 7 and 9 occupied by what? They are occupied by the nitrogen. And pyrimidines may first and third. So here you can see this is first, this is third, this is 7, this is 9, and this is not, this all are carbon. Okay, the remaining ones are the carbons. Okay, ये कार्बन पे यहाँ पे NH2 ग्रुप है, यहाँ पे डबल बॉन्ड O है. ये स्ट्रक्चर्स याद करने बहुत ज़रूरी है. pyrimidine, here NH2 group, here double bond O, double bond O, double bond O, double bond O. See, this is a structure of adenine, this is a structure of guanine, this is a structure of cytosine, uracil, and this one is thymine. So, these are all the nitrogenous bases. Okay. Now, नाइट्रोजिनस बेस हमने न्यूक्लियोटाइड के 3 कंपोनेंट तो देख लिए. वन इज शुगर, अनदर वन इज फॉस्फोरिक एसिड, एंड लास्टली इट इज नाइट्रोजिनस बेस. नाउ हाउ दिस कंपोनेंट्स विल बी लिंक्ड, इनका लिंकिंग कैसे होगा ताकि ये एक पूरा न्यूक्लियोटाइड का मॉलिक्यूल बने. सो वी विल सी द लिंकिंग ऑफ द न्यूक्लियोटाइड कंपोनेंट. A nitrogenous base is joined to the sugar molecule by glycosidic bond. So पहला किसका bonding होगा? First thing is a sugar molecule binding with a nitrogenous base. Very important students, ये याद रखना ज़रूरी है. पहले क्या होगा? The first step is binding of a sugar molecule with a nitrogenous base. And ये जो bond बनता है उनके बीच में, that bond is called as the glycosidic bond. So with the help or by glycosidic bond, a sugar molecule is linked with the nitrogenous base. Okay, linking of the sugar with the nitrogenous base, ये जो component बनता है उसको न्यूक्लियोसाइड बोलते हैं. When a sugar molecule is linked with a nitrogenous base, the structure formed is called as a nucleoside. Very important terminologies याद रखनी ज़रूरी है. Sugar plus nitrogenous base is called as a nucleoside, और उनके बीच में का जो bond है उसको बोलते हैं glycosidic bond. Okay. Now ये bond बनता किसके बीच में है? So this bond is formed between the first carbon of the sugar and the nitrogen at position 1 in case of pyrimidine and at position 9 in case of purine. Okay. What is the first step? Binding of the sugar with nitrogenous base. Okay. By which bond? Glycosidic bond. So ये bond बनता किसके बीच में है? Always it will be the first carbon of the sugar. Sugar का first carbon will bind with the first nitrogen in case of pyrimidine or the 9th nitrogen in case of the purine. Very important. Sugar का first carbon binding with the nitrogen at position 1 in case of the pyrimidine and nitrogen at the position 9 in case of the purine. The compound formed is called as the nucleoside. Okay, ये component क्या बना? Nucleoside. Now, a nucleoside with a ribose sugar is known as a ribonucleoside. जो न्यूक्लियोसाइड में राइबोस शुगर है, इट इज़ कॉल्ड एज राइबोन्यूक्लियोसाइड और सिंपली राइबोसाइड. एंड अ न्यूक्लियोसाइड विद डीऑक्सीराइबोस शुगर इज कॉल्ड एज डीऑक्सीराइबोन्यूक्लियोसाइड और डीऑक्सीराइबोसाइड. सो अभी क्या बना? न्यूक्लियोसाइड. बट वी आर टॉकिंग अबाउट व्हाट? न्यूक्लियोटाइड. सो अभी लास्ट कंपोनेंट ऐड करना बाकी है. विच कंपोनेंट इज रिमेनिंग? फॉस्फेट. सो नाउ when phosphate is attached to this nucleoside, what is nucleoside? Sugar plus nitrogenous base. So when phosphate is attached to nucleoside, the component formed will be now a nucleotide. So a nucleoside, it binds with the phosphate group at 5th position by an ester bond to form a nucleotide. very important. जो न्यूक्लियोसाइड है उसका 5th position. When we talk about 5th position, I mean the 5th position of the शुगर, किसकी फिफ्थ पोजीशन? शुगर की, नहीं के नाइट्रोजिनस बेस की। सो शुगर का जो फिफ्थ पोजीशन है दैट विल बाइंड विद द फॉस्फेट ग्रुप, एंड वो जो बॉन्ड रहता है दैट इज कॉल्ड एज एन एस्टर बॉन्ड. So binding of the phosphate at the fifth position of the nucleotide gives the formation of the nucleotide, and the bond is the ester bond. So nucleotide with ribose sugar, it is called as ribonucleotide or only ribotide, and a nucleotide with deoxyribose sugar is called as deoxyribonucleotide or deoxyribotide. See students, this is a nucleoside and a nucleotide. This is a sugar and to this this is a sugar and ये sugar से nitrogenous base attached है, so it is nucleoside. And here to sugar, this is a nitrogenous base and this is phosphate, so this is called as a nucleotide. Now जो दूसरा diagram है, here you can see the components of the DNA. See, this is first, second, third, fourth and fifth position, okay, and first position is occupied, first carbon is linked with what? Nitrogenous base, okay. So अगर purine है तो ninth position, and see students, this fifth position, this is the fifth carbon, it is occupied by what? It is occupied by the phosphate group. First carbon occupied by nitrogenous base, fifth carbon occupied by the phosphate group. So these are the various nucleotides. This is with a adenine, this is with a thymine. Thymine ho to which carbon? First carbon. This is cytosine and this is the guanine. So this is the structure of the various nucleotides. Now कौन से nucleotides and nucleosides बनते हैं, we will see that. So first of all the nucleosides. कितने nitrogenous bases है? Total 5 nitrogenous bases. Out of 5, 4 will occur in DNA and 4 will occur in RNA. So क्या कौन से components बनते हैं? We will see that. So when adenine is bind with a ribose sugar, it is called as a adenosine. We are talking about nucleosides. Nucleosides में क्या होता है? Sugar and nitrogenous base. Same way, when adenine is bind to deoxyribose sugar, it is called as deoxyadenosine. Then is guanine, which binds with ribose, it is called as guanosine. Guanine binding with deoxyribose, it is called as deoxyguanosine. Cytosine binding with ribose, it is called as cytidine. Cytosine binding with deoxyribose, it is called as deoxycytidine. Thymine will bind with only deoxyribose. Thymine किस में present है? सिर्फ DNA में. So उसका binding of course किसके साथ होगा? Deoxyribose के साथ. It will never bind with the ribose sugar. So जो structure बना, that is called as deoxythymidine. Same way uracil binds only with the ribose. It won't bind with the deoxyribose. So uracil with ribose, it is called as the uridine. So these are all the nucleosides. Now जब nucleosides के साथ phosphate group add हो जाता है, what is it called as? It is called as a nucleotide. So adenosine binding with the phosphate, it is called as adenylic acid. Deoxyadenosine binding with phosphoric acid, it is deoxyadenylic acid. Guanosine binding with phosphoric acid, guanylic acid. Deoxyguanosine binding with the phosphoric acid, it is called as deoxyguanylic acid. Cytidine binding with the phosphoric acid, it is called as cytidylic acid. Deoxycytidine binding with the phosphoric acid, it is called as deoxycytidylic acid. Deoxythymidine binding with phosphoric acid, it is called as deoxythymidilic acid, and uridine binding with phosphoric acid, it is called as the uridilic acid. Students, this is very important. किसके binding से क्या component बनता है, that is very important. Okay, याद रखने के लिए क्या है, जब न्यूक्लियोसाइड हो तो at the end it will be INE, okay, and न्यूक्लियोटाइड्स में क्या होता है, एसिड फॉर्म होगा. So that is important, students. Now we are talking about nucleic acids. Nucleic acids यानी पॉलीन्यूक्लियोटाइड. पॉलीन्यूक्लियोटाइड यानी बहुत सारे न्यूक्लियोटाइड जब जॉइन होते हैं. अब तक तो हमने न्यूक्लियोटाइड के अंदर का लिंकेज होगा. नाउ हाउ द टू न्यूक्लियोटाइड्स विल बी अटैच्ड विथ ईच अदर, so that results in the formation of a dinucleotide. So two sequential nucleotides join through phosphodiester bond and it form a dinucleotide. So what is a dinucleotide? When two nucleotides they join by the phosphodiester bond, the component form is called as a dinucleotide. Now question arises, ये bonding होगा कैसे? So union occurs between the third carbon of the sugar in one nucleotide to the fifth carbon of the sugar in the other nucleotide. Very important. एक nucleotide का third carbon binds with the phosphate which occurs at the fifth carbon of the other nucleotide. Listen very carefully, students. The nucleotide, third carbon of the sugar in one nucleotide, it binds with the fifth carbon of the sugar which is occupied by the phosphate at the of the other nucleotide. That is, phosphate component of one nucleotide is joined by phosphodiester bond to the sugar component of the adjacent nucleotide at the third carbon. ओके, फॉस्फेट कंपोनेंट किसके कौन से कार्बन पे है, 5 कार्बन. सो दैट कार्बन विल बाइंड विद द थर्ड कार्बन ऑफ द नेक्स्ट न्यूक्लियोटाइड. सो दिस इज हाउ अ डाइन्यूक्लियोटाइड इज फॉर्म्ड. नाउ व्हाट आर न्यूक्लिक एसिड्स? दे आर पॉलीन्यूक्लियोटाइड. सो व्हेन many nucleotides are joined in this manner, what structure will be formed? A polynucleotide. So when many nucleotides unite, a polynucleotide chain is formed. Say for example, एक हम एग्जाम्पल लेते हैं कि फर्स्ट जो न्यूक्लियोटाइड है उसका थर्ड है, इट विल बाइंड विद द फिफ्थ ऑफ द अदर. अभी ये जो अदर है उसका थर्ड बाकी है, वो किसके साथ ज्वाइन हो जाएगा? फिफ्थ दूसरे का. सो इन दिस वे व्हेन मेनी न्यूक्लियोटाइड्स ज्वाइन इट फॉर्म्स अ पॉलीन्यूक्लियोटाइड. There is one polynucleotide chain in the structure of RNA where there are two polynucleotide chains in the structure of DNA. RNA में एक ही polynucleotide chain होती है and DNA में 2 polynucleotide chain होती है. We will study the detailed structure of the DNA and the RNA in your 12th standard. Now see, this is what is given in your textbook. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases like adenine, guanine, cytosine, uracil and thymine. When found attached to sugar, they are called as nucleosides. When what? So nitrogenous bases plus sugar, what is it called as? Nucleoside. If phosphate group is also found esterified to the sugar, they are called nucleotide. अभी इसमें जब फॉस्फेट ग्रुप अटैच हो जाता है तो उसको हम क्या बोलते हैं? न्यूक्लियोटाइड्स. वी हैव स्टडीड देम इन डिटेल इन प्रॉपर वे. एडिनोसाइन, गुआनोसाइन, थाइमिडिन, यूरिडिन एंड साइटिडिन आर न्यूक्लियोसाइड्स. And adenilic acid, thymidilic acid, guanylic acid, uridilic acid, cytidilic acids, they are the nucleotides. Nucleic acids like DNA and RNA, it consists of nucleotides only. DNA and RNA, they function as the genetic material. These are the diagram given in your book. This is a purine, this is a pyrimidine. Only two of them are mentioned. One is adenine and another one is uracil. This is first, third, seventh and ninth, and this is first and this is the third position. Now this is adenosine. Adenosine is a ribose sugar. This is the first carbon having a adenine, and again this is adenylic acid. Difference क्या है? Look at it carefully. So this is the first carbon जहाँ पे adenine है, and this is adenosine. यहाँ पे first carbon adenine, fifth carbon पे phosphate, that is called as the adenylic acid. And this is uridine जहाँ पे first carbon पे uracil है. So these are nitrogenous bases, these are nucleosides, and this one is a nucleotide. Nucleic acids, they are the other type of macromolecule one would find in the acid insoluble fraction of any living tissue. They are polynucleotide along with the polysaccharides and polypeptide. They comprise the true macromolecular fraction of a living tissue or cell. The building block is nucleotide. A nucleotide has three chemically distinct components. We have studied that. One is a heterocyclic compound called as nitrogenous base. Second one is a monosaccharide, which is a pentose sugar ribose or deoxyribose. And third is phosphoric acid, or also called as the phosphate group. The heterocyclic compounds in the nucleic acids are nitrogenous bases named adenine, guanine, uracil, cytosine, thymine. Adenine and guanine are substituted purines, while rest are pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose, a monosaccharide pentose, or 2-deoxyribose. 2- क्यों लिखा है? क्योंकि सेकंड कार्बन के ऊपर जो OH ग्रुप है उसका ऑक्सीजन निकल गया है. A nucleic acid containing deoxyribose is called as deoxyribonucleic acid, while which contains ribose is called as the ribonucleic acid. So that is regarding the nucleic acids. Now let us begin with the new topic, that is primary and secondary metabolites. So first of all, what are metabolites? So living organisms में there are organic compounds which are constantly used during the metabolic reactions. There are organic compounds which are constantly utilized during the metabolic reactions. कोई भी लिविंग ऑर्गेनिजम्स में मेटाबॉलिक रिएक्शंस कंटिन्यूसली बॉडी में होते रहते हैं. Those metabolic reactions are of two types, catabolic and anabolic. Anabolic यानी जिसमें कुछ सिंथेसिस होता है, and catabolic यानी जिसमें कुछ ब्रेकडाउन होते हैं. सो कंटिन्यूसली लिविंग ऑर्गेनिजम्स में केमिकल रिएक्शंस होते रहते हैं. सो ये मेटाबॉलिक रिएक्शंस में ऑर्गेनिक कंपाउंड्स आर यूज्ड. सो दिस ऑर्गेनिक कंपाउंड्स आर कॉल्ड एज व्हाट? मेटाबोलाइट्स. The most exciting aspects of chemistry deals with isolating thousands of compounds, small and big, from living organism, determining their structure and if possible synthesizing them. So जो most important challenge है chemistry की क्या है कि to isolate the various compounds which are present in the living organisms. These compounds may be smaller, they may be bigger, etc. Not only that, we need to determine their structure and if possible to synthesize them. Thousands of organic biomolecules like monosaccharides, fatty acids, glycerol, lipids, amino acids, nitrogenous base, phospholipids, nucleotides, they are present in the cell. These organic compounds are constantly utilized in various metabolic, that is anabolic and catabolic reactions occurring in the cells. And hence these biomolecules are called as metabolites. So as these biomolecules are constantly utilized in various metabolic reactions occurring in the cell, what are they called as? They are called as the metabolites. In animal tissue, all the categories of organic compounds are found. So ये जो भी ऑर्गेनिक अभी तक जो भी बायोमोलिक्यूल्स हमने स्टडी किए, लाइक शुगर यानी कि मोनोमेरिक यूनिट जैसे शुगर, एमाइनो एसिड्स, फैट्स, नाइट्रोजन बेसेस, न्यूक्लियोसाइड्स, न्यूक्लियोटाइड्स, ये सब कंपोनेंट एनिमल टिश्यू में प्रेजेंट है. And what are they called as? They are called as the primary metabolites. What are they called as? Primary metabolites. These are required for basic or primary metabolic processes like respiration, lipid and protein metabolism, etc. Now, इसको क्यों primary metabolites बोलते हैं? These are called as primary metabolites because they are required for the primary metabolic activities या अपने growth and development के लिए compulsorily these all components need to be present and hence they are called as the primary metabolites. But when one analyzes fungal, plant and microbial cell, one would see thousands of compounds other than these primary metabolites. So, if we examine the plant cell, fungal cell or microbial cell, along with these primary metabolites, we will find other thousands of compounds other than the primary metabolites. प्राइमरी मेटाबोलाइट्स तो हो गए, लेकिन उसके अलावा दूसरे thousands of compounds they are present in the plant, fungal and microbial cells. Examples of such other compounds are alkaloids, flavonoids, rubbers, essential oils, antibiotics, colored pigments, scents, gums, spices, etc. So, these components are called as the secondary metabolites. इसको क्या बोलते हैं? Secondary metabolites. Now, difference दोनों का क्या है? Secondary metabolites plant में, fungal में, microbial cell में produce तो होता है, लेकिन ये components का वो particular organism के लिए क्या usefulness है? They don't have basically they are not directly related to growth and development of the organism. जिनमें ये component present है, इनमें ये component growth या reproduction या development से उसका कुछ भी लेना देना नहीं है, फिर भी उनमें वो present है. जबकि प्राइमरी मेटाबोलाइट्स क्या होते हैं? दे आर डायरेक्टली इन्वॉल्व इन द ग्रोथ एंड डेवलपमेंट ऑफ एन ऑर्गेनिज्म. बट जो सेकेंडरी मेटाबोलाइट्स है, जो ऑर्गेनिज्म में प्रेजेंट है, उनमें उनका कोई डायरेक्ट रिलेशन ग्रोथ या डेवलपमेंट से नहीं है. सो व्हाट आर दे कॉल्ड एज सेकेंडरी मेटाबोलाइट्स? They are not involved in primary metabolism and seem to have no direct function in growth and development of the organisms. While primary metabolites have identifiable functions and play the known roles in normal physiological processes, we do not at moment understand the role or functions of all the secondary metabolites in host organism. Host organism यानी जिनमें वो present है, उनमें सेकेंडरी मेटाबोलाइट्स क्या करते हैं उसका कोई भी अभी तक फंक्शन टिल मोमेंट अपने को उनका क्या फंक्शन है होस्ट ऑर्गेनिज्म यानी जिनमें वो कंपोनेंट प्रेजेंट है उनमें वो क्या फंक्शन कर रहे हैं इट इज़ नॉट येट नोन. हावेवर मेनी ऑफ देम आर यूजफुल टू ह्यूमन वेलफेयर. हाँ, वो जो सेकेंडरी मेटाबोलाइट्स है वो ह्यूमन बींग्स को हेल्पफुल है, लेकिन जो प्लांट में एग्जिस्ट करते हैं, जो माइक्रोब्स में उन लोग प्रेजेंट है या जो फंगस में प्रेजेंट है, उनमें उनका क्या फंक्शन है वो नोन नहीं है. एग्जाम्पल, रबर इज़ यूजफुल, ड्रग्स, स्पाइसेस, सेंट्स, पिगमेंट्स, दे आर ऑल यूजफुल टू ह्यूमन बींग्स. Some of the secondary metabolites also have the ecological importance. See students, these are some secondary metabolites. Pigments like carotenoids, anthocyanins, etc. Alkaloids like morphine, codeine, etc. Terpenoids like monoterpenes, diterpenes, etc. Essential oil like lemongrass oil, etc. Toxins like heparin, ricin, etc. Lectins like concanavaline, etc. Drugs like vinblastine, curcumin, etc. and polymeric substances like rubber, gum, cellulose, etc. These are the examples of some of the secondary metabolites, of which many are useful to the human beings. We will study the importance of some of the secondary metabolites like lectins. They are used as pesticides, fungicides, as a drug for cancer, antiviral drug, etc. So हम बोल सकते हैं कि lectin though it is produced by the प्लांट्स और माइक्रोब्स और फंजाई, बट वो अपने लिए हेल्पफुल हो सकते हैं। प्लांट के लिए या फंगस के लिए या माइक्रोब्स के लिए कैसे यूज़फुल है या यूज़फुल है भी या नहीं, अभी तक वो नोन नहीं है। Alkaloids are used as diet ingredients, supplements and pharmaceuticals and in other applications in human life. They also act as local anesthetics, anti-tumor agents, antibacterial, antimalarial, painkillers, etc. Therapeutic uses, that is medicinal uses of terpenoids include antimicrobial, antifungal, antiviral, antihyperglycemic, anti-inflammatory, antioxidants, antiparasitic, immunomodulatory, and they also act as a skin permeation enhancers. And lastly, essential oils, they are used to ease stress, boost mood, relieve the pain from headaches and migraines, get better night sleep, quell or stop nausea, and they also even repel the insects. So these are some of the usefulness of the secondary metabolites to the human beings. I end my lecture here. In the next lecture, we will be dealing with the enzymes and some other topics. Thank you, students.